Hello again. In the previous video, we talked about how the promoter plays its role as a starting point of transcription. The other point that we're going to look at in this slide is the ending, how the polymerase system, the transcription system, recognizes where to stop. And this is this occurs by the fact that towards the end of the genes, there are regions what you call as inverted repeats. They are inverted repeats. That means the characteristic of inverted repeats is that they will be complementary and they can form what you call as hairpin structure. This is sometimes called hairpin structure. This is also called stem and loop. Different books use different terms. And because I mentioned also, I use the analogy of sewing. When you sew, when you start pulling it through the thread through through the cloth, and sometimes it gets stuck. And when it gets stuck, you cannot move. So it's the same thing happens here. If the polymerase that it goes along the RNA, and then if this RNA loops, if you can see this loop is still within the mRNA, then it sort of stalls the polymerase. And because the polymerase is now stalled, it gives a signal for it to say that, okay, this is the end of transcription, right? And you can also see at the ends here, the ones that's holding the RNA and the DNA are just this AU rich region. What's the significance of that? AU is less strong than GC. Why? Because AUs have only 2 hydrogen bonds each. GC has 3 hydrogen bonds each. So it's less. The bonds that's holding the RNA and this DNA is not as strong as the GC bond. So that's why this is also called the RNA-DNA hybrid region. And again, because RNA polymerase is stalled, so it gives a signal for it to just dissociate, and that marks the end. Another way to stop transcription is also the involvement of Rho factors. Again, these Rho factors come ahead of the ribosome sometimes, but it recognizes. You can see that there's also looping here that is recognized by the Rho factors, and the Rho factors again. If you read papers on Rho factors, it's quite elaborate. It also involves some structural changes in the Rho factors themselves. You know, they form a sort of a loop kind of thing, and then again, the important point is that it gives a signal to the polymerase that this to stop transcription. These are just some information comparatively between what happens between a prokaryote and eukaryote. The overall process is similar, it's almost similar, but they use different types of proteins. And of course, in the eukaryote, if you remember the first, on one slide it says, you know, you need to process it. There are removal of introns, there's capping, there's adding A. So you know, so that is much more complicated in the eukaryote system. So that's why when it's in this class, in this first year course, the main focus is in the prokaryotic system. Okay, so I'll see you in the next video when we talk about translation. Bye bye.